What's up, microbiologists, biologists, and fellow YouTube enthusiasts? Welcome to another edition of Purple Bear Biology. In this episode, what we're going to discuss is three main topics. One, why and how we identify bacteria down to a genus or species. Two, the importance of enzymatic function in identification of bacteria. And then three, what general tests do we use to identify gram-positive cocci like Staphylococcus and Streptococcus? I hope you're all prepared to have your minds blown in how you identify bacteria. Let's get started. Imagine you were in a lab and you held up this agar plate. You have 5 unknown bacteria on there, and you have no idea which is which. How would you determine which bacteria is which on the plate? Also, why would you want to know what genus and species you have in the first place? In microbiology lab classes at the university level, we often start with learning how to use a microscope and then progress to isolating bacteria. Once they are isolated, we focus on staining them so that they are visible and we can learn about their shape, arrangement, and morphological features. We can also explore the biological growth factors of microbes to determine the minimalist or enriched media that we need to grow the bacteria in the lab. While all of those features can be useful in identification of a bacteria, none of them individually would be enough to definitively identify most bacteria to species. The reason for that is the similarities between those features across different microbial groups. So what can we do to begin to identify bacteria and separate them into major biological groups? We often use the microbe's ability to make a particular enzyme. Recall that an enzyme is a specialized protein that helps chemical reactions proceed. Enzymes often enable bacteria to live in unique environments and deal with specific biological conditions that a microbe may find themselves in. This may be to survive a certain temperature range, operate at a particular pH, or even catabolize compounds that would otherwise be toxic. Groups of bacteria are often divided into different genera and are grouped together based on very similar characteristics. These characteristics are often the different enzymes that they have. This can be hugely influential and beneficial for identification because it enables us to present chemical scenarios to microbes and see how the bacteria respond. Now that's all pretty cool, but why do we care about the identification of the bacteria in the first place? Why do I care when I look at these plates that I have Staphylococcus or Streptococcus? The most direct answer would be to determine if the bacteria that is present is of need of concern. In other words, is it pathogenic? And then, if it is dangerous, we could use the identification to determine the most appropriate treatment and which antibiotics would be most effective. In our exploration today of enzymatic function, we are going to focus on separating two major groups of gram-positive bacteria, Staphylococcus and Streptococcus species. Both groups contain pathogenic and relatively non-pathogenic species. They present us with an opportunity to explore biochemical identification in order to determine which species are present and whether or not there's a need for concern. We will start by talking about the different tests and results we can obtain from those tests in the different groups, and then you are going to use the results in a series of biochemical tests to determine the identification of 5 unknown bacteria. Because both of these groups have multiple species, and we only have a limited amount of time available, for the purpose of this exploration, we are going to limit our bacterial species to 2 staph species and 3 Streptococcus species. Okay, so now let's talk about identifying some bacteria. The first step in identifying bacteria would be to perform a gram stain. Remember that normally a gram-positive bacteria would be purple and a gram-negative bacteria would be pink. All of the bacteria that we are going to discuss are gram-positive. So let's move on to the next identification step, the Catalase test. Catalase is an enzyme produced by staph species that enable them to survive in the presence of hydrogen peroxide. The result of the catabolic reaction is that catalase enzyme breaks the hydrogen peroxide down into water and oxygen. If we look at this with the bacteria present inside of the peroxide solution, the oxygen will produce bubbles. Staph will produce bubbles for a positive catalase test, while streps would not produce bubbles or few bubbles. Let's take a look at a positive result. In this video clip, I have placed hydrogen peroxide and added a little bit of crystal violet to stain the solution all on a glass slide. The result is a slightly purple stained peroxide solution under the microscope at 40 times total magnification. Ooh, look at all those bubbles. So this result would tell us that we have a staph species and not a strep species. This means that the catalase enzyme is present and is actively breaking down the hydrogen peroxide, and the oxygen is producing the bubbles. Okay, now let's look at the same test to see what a negative result would look like. Again, we have a hydrogen peroxide solution with a little bit of crystal violet applied at 40 times total magnification. You can see that there are no bubbles present. So this result would tell us that we are working with the Streptococcus species because it does not have the catalase enzyme. Now that we have separated them into two separate genera that we are looking at today, we can try to identify the species that we are working with. Let's start with our positive catalase result that identified our Staphylococcus species. The next step would be performing a Coagulase test to determine if we have a pathogenic Staph aureus or the mostly non-pathogenic Staphylococcus epidermidis. We would use this Coagulase test. This test uses rabbit plasma. A positive result is noted by a coagulation of the plasma and can be visibly seen as clumps that appear on the slide. So let's take a look at a positive result first. Notice that you start to see small clumps appear inside the solution. This would tell us that we have a positive result and that the pathogen we're looking at is Staphylococcus aureus. Let's compare that with a negative result. Here we have the rabbit plasma on the slide. I've added a little bit of stain to turn it red, and we're applying bacteria to see if it'll clump. Notice that there are no little clumps on the slide as we tap the bacteria in there. This would tell us that this is not Staphylococcus aureus, but instead is Staphylococcus epidermidis. This is a common staph that you would find on your skin. In addition to those two tests to confirm our result, by growing our bacteria on a mannitol salt agar plate, we can determine whether or not we have Staphylococcus aureus or Staphylococcus epidermidis. The salt agar plate is both selective and differential. The high salt concentrations select for staph species, and there is a pH indicator that turns yellow when Staphylococcus is grown on it. And believe it or not, that's it for identifying these two staph species we're looking at today. So let's move on over to our Streptococcus species and talk about how we identify them. To begin to identify Streptococcus species, we use something called their hemolytic pattern. This is essentially how the bacterial colonies will look when they grow on a sheep blood agar plate. There are three different types of hemolytic patterns that can be exhibited. We have beta hemolysis, or complete hemolysis, that is typically noted by the plate turning clear around the bacterial growth. In this pattern, the red blood cells are actually being ruptured as the bacteria use them for a nutrient resource. The second pattern we see is alpha hemolysis. This pattern tends to turn a green tint and is sometimes called incomplete hemolysis. The red blood cells are not ruptured in this pattern. Instead, the bacteria actually produce chemicals that cause the hemoglobin to oxidize and turn green. The last pattern is gamma hemolysis, and it's also known as non-hemolytic. We can see that there is growth, but no red blood cell disruption. Sometimes there's a temptation to skip the Catalase test when trying to determine Staphylococcus and Streptococcus species. Students will jump right to the hemolytic pattern and try to identify it as a Streptococcus species. You must never do this because other bacterial species, including staphs, can exhibit hemolytic pattern. In general, we never want to identify using just one result, and that includes hemolysis. Once we have identified the hemolytic pattern, we can begin to develop our suspicions about which microbe we have, but we have to confirm those suspicions with a series of secondary tests. For beta hemolysis, it actually exhibited by several strep species, and we group them into different groups like group A strep, group B strep, and so on. To test if we have group A, we test for antibiotic sensitivity to bacitracin. A zone of inhibition around the antibiotic disc would confirm our identification to be group A Streptococcus species, and in this case, Streptococcus pyogenes. However, if there is no zone is not susceptible to bacitracin, then we could follow it up with a test called a CAMP test. In this example, you can see that we've taken Staph aureus and streaked it in a straight line. We did the same thing with the Streptococcus species we're trying to identify. A positive result for the CAMP test would show us this arrowhead-like formation in the hemolytic pattern. This would confirm that it's actually a group B Streptococcus species. But what happens if none of those tests are positive? Well, there's one more test we could run. The last test to test for our Streptococcus species that are exhibiting a beta hemolysis pattern would be to grow them in an enterococcal broth. This media starts off a golden color, and if a group D Streptococcus faecalis is present, it will turn pitch black. If all of those results are negative, then we would have to list this as an unknown beta hemolytic Streptococcus species. Coming back to our diagram, alpha hemolytic streps could be Streptococcus pneumoniae. To confirm this prediction, we would need to test for antibiotic sensitivity to optocin. Like the bacitracin antibiotic, a zone of inhibition around the antibiotic optocin disc would confirm our identification as Streptococcus pneumoniae. If the bacteria are not susceptible to this antibiotic, then we could only identify it as an unknown alpha hemolytic Streptococcus species and would need further testing to determine the exact identity. You could also do an enterococcal broth test to see whether or not this was Streptococcus faecalis exhibiting a different hemolytic pattern. The last hemolytic pattern is the gamma hemolysis, and then again we would want to test using an enterococcal broth inoculation. If it turned black, we confirm that this was Streptococcus faecalis. Otherwise, we would list it as an unknown gamma hemolytic strep. Okay guys, so hopefully now you have some idea of why we identify bacteria down to bacterial and species, how we can use enzymatic function for those identification processes, and then the series of steps that we would take in order to try to identify some gram-positive cocci, like Staphylococcus and Streptococcus, starting off using a gram stain to identify that they're gram-positive in the first place, then using a Catalase test to distinguish between staphs and streps, and then on the staph side, using a Cocciase test to determine what is Staph aureus, and then using an MSA plate in order to confirm those results. And then over on the strep side, we use hemolytic patterns as well as some secondary tests to confirm our results and make sure that we know which particular Streptococcus species we're talking about. All of that you can hopefully apply and use. For my microbiology students, I'm going to give you some examples of some unknowns to try to figure it out, and I hope y'all have yourselves a wonderful day. If you liked this video and found it useful, make sure that you hit the like button and tell me about it down below, and also hit the subscribe button so you can see more videos like it. Y'all have yourselves a wonderful day, and that's it for this episode of Purple Bear Biology.